Hello and welcome students to the SRBPS virtual classroom program. I am your biology educator Prerna Rathi. I hope you are all making complete use of our YouTube channel. Now let us come and see what I have got for you today. Here we are yet again after discussing all the details of micromolecules and macromolecules. Now we have the enzymes part of biomolecules. Children, it's a very important topic in this chapter. Let us discuss what are enzymes. In the mouth, there are various enzymes present in the stomach, various enzymes present in the small intestine. And what are these enzymes? Enzymes are biocatalysts. These are those substances, those chemicals which enhance the rate of बायोकेमिकल रिएक्शंस. जो लिविंग वर्ल्ड के रिएक्शंस हैं उनके रेट ऑफ रिएक्शन को इंक्रीज कर देता है विदाउट एक्चुअली पार्टिसिपेटिंग इन द रिएक्शन. Fine. Let us discuss the basic characteristics of enzymes. Enzymes are proteins that will catalyze the biochemical reaction. That's why they are known as biocatalysts. Most of them are proteinaceous in nature except for a few which are RNA in nature, and those who are RNA in nature are known as ribozymes. We don't call them enzymes, we call them ribozymes. Remember, all the enzymes are always substrate specific. They work for a particular substrate. They cannot be used for any other substrate. For example, salivary amylase जो हमने क्लास 10th में किया था वो सिर्फ माउथ में प्रेजेंट स्टार्च को ब्रेक कर रहा था. उसके अलावा प्रोटीन और किसी कार्बोहाइड्रेट, किसी लिपिड, किसी चीज़ के साथ वो कोई भी इंटरफेरेंस नहीं कर रहा था. Each enzyme always requires a specific temperature and pH to show its best activity. Below or above that, its activity would be deviated from its best performance. Enzymes accelerate the rate of reaction by reducing the activation energy. Normally ये जो biochemical reactions होते हैं बेटा, इनकी जो activation energy इतनी high होती है कि without the presence of an enzyme वो activation energy की value को gain कर पाना ही nearly impossible होता है. Now let us discuss what are the similarities between the enzymes and the inorganic catalysts that we have learned so far. The catalysts remain unchanged at the end of the reaction. So enzymes भी unchanged रहते हैं और जो inorganic catalyst हैं वो भी unchanged रहते हैं. They are required in a far lesser quantity as compared to the substrate. रेट ऑफ रिएक्शन को फास्ट कर देते हैं बाय डिक्रीजिंग द थ्रेशोल्ड और द एक्टिवेशन एनर्जी. दे डू नॉट ऑल्टर द इक्विलिब्रियम ऑफ अ रिवर्सिबल रिएक्शन. अगर रिएक्शन रिवर्सिबल है तो इसका मतलब ये नहीं है कि हमेशा रिएक्टेंट से प्रोडक्ट ही बनता रहेगा जब तक वो एंजाइम प्रेजेंट रहेगा. इफ द एनर्जी इज इनफ फॉर द प्रोडक्ट टू बी कन्वर्टेड बैक इनटू रिएक्टेंट देन दैट विल हैपन. They form very short-lived complexes with the substrate which are known as enzyme substrate complexes. What are the differences? All enzymes are proteins and they have a very complex molecular organization, whereas inorganic catalysts like Mn2 positive, Mg2 positive, KMNO4, K2Cr2O7 बहुत बेसिक स्ट्रक्चर के इनऑर्गेनिक कैटलिस्ट हैं. एंजाइम्स जो होते हैं वो एक स्पेसिफिक रिएक्शन के लिए काम करते हैं. दे आर सबस्ट्रेट स्पेसिफिक, whereas inorganic catalyst children are not substrate specific. Understood? Enzymes can be regulated. The working of an enzyme can be regulated, but we cannot regulate the inorganic inorganic catalyst. The enzymes are more sensitive to the pH and temperature changes, because they are proteins. They get denatured and they either enhance their activity or reduce their activity due to the pH and the temperature change, whereas it brings about nearly negligible change in inorganic catalysts. Let us now classify the enzymes into various categories. This classification has been given by IUB, that is the International Union of Biochemists. It has categorized enzymes into 6 categories. Let us discuss all the 6 one by one. The first category is oxidoreductase, those enzymes which cause oxidation and reduction reaction. For example, there are two substrates S and S dash. One would be oxidized and one would be reduced. We have already read about redox reactions in class 10th. Then there are from S to S dash, that is why it has formed a complex S dash J. Hydrolases वो enzymes होते हैं which catalyze the breakdown of larger molecules into smaller molecules with addition of water. We add a water molecule and cause the breakdown of larger molecule. Then we have lyases. Lyases क्या करते हैं? They work on specific covalent bonds and break them. For example, अगर इसमें आप देखेंगे there is C2X5, तो ये X CX और CY वाले bond पे ही काम करेगा, CC bond पे काम नहीं करेगा, and will lead to formation of a C double bond C. Then we have isomerases, which do not break anything, which do not make anything, but they form isomers, the molecules which are structurally different, but molecular formula is same, the same molecular formula but different structural formula. Then we have ligases. Ligation का मतलब होता है join करना. So ligases are those enzymes which cause ligation or joining of two or more molecules. Let us now discuss how they work on chemical reactions. Chemical compounds undergo two types of changes whenever there is a chemical reaction. Either it is a physical change or a chemical change. And always the rate of change, whether it is a physical or chemical change, refers to the amount of product formed. And this rate of reaction is equal to delta P by delta T. Delta P is the little amount of product that is formed, delta T in little time, okay, because we are taking very small amounts, that's why we are writing delta P by delta T. And this rate is also known as the velocity of the reaction. The rate of the reaction is directly influenced by the temperature. The general thumb rule that we use for enzymes is that, children, the rate doubles or decreases by half. Double up हो जाएगा अगर temperature 10 degree Celsius reactions proceed at a much faster rate. For example, carbon dioxide and water का conversion in the presence of carbonic anhydrase into carbonic acid is accelerated about 10 million times in the presence of this enzyme. So it accelerates the reaction, it increases the velocity. Then there are various metabolic pathways, and metabolic pathways क्या होते हैं? For example, glycolysis, photosynthesis, ये सब metabolic pathways है. ये catabolic भी हो सकते हैं, anabolic भी हो सकते हैं. And they are a multi-step reaction, and at every step a different enzyme is required. Why beta? Because at every step a new substrate is formed, and we know that enzymes are substrate specific. For example, glucose को अगर हमें pyruvate में convert करना है, this is a 10-step reaction. Glycolysis एक 10-step reaction है, और हर एक स्टेप पे एक डिफरेंट एंजाइम यूज होता है. फाइन. हाउ डू दीज एंजाइम फंक्शंस? एंजाइम्स कैसे काम करते हैं? ये जो एंजाइम होता है, जस्ट एक्सप्लेन ब्रीफली. Let me now explain how the enzymes will function. As we already know, enzymes कैसे function करते हैं? They bring down the activation energy, as you can see in this particular graph. This is the activation energy that is required without the presence of the level of the substrate, and this is the energy level of the product. Now with this graph only we can understand, अगर substrate का energy level higher, product का नीचे है, then it is an exothermic reaction. This energy will be released. On the other hand, अगर product का energy level high है, substrate का low है, then it is going to be an endothermic reaction, and we might have to supply energy. We actually have to supply energy from outside for this reaction to occur. Okay, children. Next, the nature of enzyme action. We've already studied how enzyme and substrate combine to form a transitionary enzyme substrate complex, which converts into enzyme product complex, which releases the product from the surface of the enzyme. Now let us discuss the effect of various factors like temperature, pH and substrate concentration on the enzyme activity. Now temperature, we have already discussed this. Temperature के साथ क्या होगा? enzyme activity increase होती जाएगी, but only up till a certain point. इसके बाद यहाँ पर ये maximum activity show करेगा at a particular temperature. इसके बाद अगर आपने further temperature increase किया तो enzyme activity will drop. Similar is the graph for pH as well. Just a second. The pH graph is here, which is similar to the enzyme graph, the temperature graph. It increases first and then decreases. There is a particular pH, where a specific pH where the enzyme activity or the velocity of the reaction is maximum. After that it will drop. Now effect of substrate concentration. देखो substrate concentration के साथ क्या होता है? हम इनिशियली जब सबस्ट्रेट कॉन्सेंट्रेशन बढ़ाते जाते हैं तो हमारा वेलोसिटी ऑफ रिएक्शन बढ़ता जाता है. सपोज यू हैव 100 एंजाइम्स, 100 कॉन्सेंट्रेशन नंबर में बता रही हूँ मैं, 100 एंजाइम्स हैं. तो जब तक आप सबस्ट्रेट कॉन्सेंट्रेशन को 100 की वैल्यू पे बढ़ा तक लेकर जाओगे तो जैसे जैसे आप सबस्ट्रेट डालोगे वैसे वैसे स्पीड ऑफ रिएक्शन Hence, after this curve is going to be a plateau curve. What will it be? It will be a plateau curve. So this is the value of Vmax, where the plateau will start. Now the half the value of Vmax is called half Vmax, and the value of substrate concentration at half the velocity is known as the Michaelis-Menten coefficient, which is very important constant for every enzyme. Now let us discuss cofactors. What are cofactors? Cofactors क्या होते हैं? ये एंजाइम को हेल्प करने वाली एंटिटीज़ होती हैं, केमिकल्स होते हैं, नॉन प्रोटीन मॉइटीज़ होती हैं. ऐसा एंजाइम जिसके साथ कोफैक्टर्स ज्वाइन करके रहते हैं, दैट एंजाइम इज़ नोन एज अ कंजुगेट और अ हॉलो एंजाइम. हॉलो एंजाइम में दो पार्ट होते हैं. एक उसका एपो एंजाइम जो इसकी प्रोटीन मॉइटी है, और इसकी नॉन प्रोटीन मॉइटी विच इज़ नोन एज द कोफैक्टर. द कोफैक्टर्स आर ऑफ थ्री टाइप्स: prosthetic groups, coenzymes, and the metal ions. Prosthetic groups are the organic compounds which are always permanently bound to the enzymes. Okay. For example, heme is the prosthetic group which works in enzyme peroxidase and catalases. Then there are coenzymes. Coenzymes सिर्फ तभी join करते हैं जब enzyme कोई activity perform कर रहा होता है. Otherwise, they do not join to the enzyme. Example of coenzymes are vitamins like NAD, NADP. Okay, they have vitamin and niacin, ये सब present होता है इनके अंदर, and they are coenzymes. And there are certain enzymes जो metal ions की presence में काम करते हैं. For example, कुछ enzymes को Mn2 positive, Mg2 positive, zinc as a cofactor की requirement होती है. So metal ions are also considered to be cofactors. What I wanted to convey here is the cofactors are three entities which help or which facilitate or which improve the catalytic function. I hope you have all understood what I taught you and you are making complete use of this YouTube channel. Until next time, take care, study hard. Goodbye, children.